We're going to solve this recent P5 paper. It's Feb March 2023. 52 variant paper. You had students, they had 1 hour and 15 minutes to solve this paper. Let's start. All right, they are saying that potatoes are underground organs made by a potato plant, Solanum tuberosum, and this figure shows this plant. So we have a plant here, and this plant is producing underground stems, which are potatoes. Now they say that they contain high quantities of starch and are very popular food. For sure. After harvesting, they are stored, and during storage, their starch content gradually decreases due to the breakdown of starch molecule into glucose. Over the time, the starch molecules they are converted into glucose. Now, a glucose assay using a Benedict's test and a calorimeter can be used to determine the concentration of glucose in potatoes. As you guys already know, what is Benedict test? It's a physical test that is used to, estimate the concentrations of reducing sugar in some of the samples, some of the prepared samples. Now, the glucose assay uses these steps. They're saying that the outer skin of potato is removed from potato, and potato is cut into small pieces. एक आलू था, उसको उन्होंने skin उतार दी. उसके बाद they cut into a small pieces and then ground into a pulp using mortar and pestle. तो उन्होंने उसके बाद उसके छोटे छोटे पीस किए. उसके बाद they use a mortar and pestle and they made a paste. एक स्लरी की तरह की चीज बना ली उन्होंने mortar pestle को यूज करते हुए. This potato pulp is filtered to remove most of the solids and obtain potato juice. So now we are obtaining potato juice out of it. An excess Benedict solution is added to potato juice. Now we're adding a lot of potato Benedict solution to its juice. Right. So now they're saying that this mixture is heating using a water bath at 90 degrees centigrade for 5 minutes. And during this 5 minutes period, copper ions in the Benedict solution, they react with glucose in potato juice to form insoluble precipitates. And the mixture is filtered to remove the precipitate. The filtrate, which is blue, contains copper ions. Did not react with the glucose in potato juice while mixture was being heated. A sample of blue filtrate is transferred to a calorimeter cuvette, and a calorimeter is used to measure the absorbance of the blue filtrate. A calibration curve is used to determine the glucose concentration of the potato juice from the measurement in the absorbance. अब उन्होंने इस प्रोसेस के अंदर एक डिफरेंट चीज ये की है कि उन्होंने ये सारा कुछ करके जब उसको वाटर बाथ में रखा और जो यहाँ पर प्रेसिपिटेट्स बन गए, बाद में उन्होंने एक कॉनिकल फ्लास्क यूज की होगी या जो भी चीज यूज की होगी, उसके अंदर एक फिल्टर पेपर लगाकर इन प्रेसिपिटेट्स को फिल्टर कर लिया. अब जो नीचे उनके पास मटेरियल आया जो फिल्टर हो चुका है, इसको उन्होंने कैलोरीमीटर के अंदर डाला है. और कैलोरीमीटर का प्रिंसिपल मैं आप लोगों को अगर नहीं अंदाजा तो मैं आपको समझा देता हूँ. इन कैलोरीमीटर वी हैव a monochromatic light. We have a monochromatic light and then we have a slit. This is monochromatic light source. and this is a slit from which a beam of light is going. And then we have a cuvette. We have a transparent tube. We have a transparent tube in which we are putting in our sample. And then we have a sensor which is receiving light that is coming out of this tube. So, bacho, if you're putting in distilled water inside this tube, we can expect that all light will pass through it. So there is nothing to absorb this light and there is everything to transmit. But as we start increasing some concentration of particular molecules inside this tube, then for sure they are going to absorb the light and less light would be transmitted. So as we increase the concentration of any particle which can hinder the light, absorbance is increased and transmittance is decreased. Right? So this is the principle of calorimeter that they are going to use in this experiment. Onwards, let's see what they're saying. Right. So this figure shows one type of a calorimeter in which we have a calorimeter tube and then we have 3 ml of sample with a blue filtrate and we have digital readout absorbance. Here we are going to read out and this is the hole to hold the calorimeter tube. Right. Now what they're saying. A clean calorimeter tube containing sample to be tested is placed inside the calorimeter and light is passed through the sample and the absorbance of light is by the sample is measured. Now he's saying that outline how calorimeters should be prepared before carrying out measurements on samples so that correct absorbance readings are obtained. Now we need to answer this question. Number one, we need to set a certain wavelength of light. Means we need a filter, actually colored filter. Remember I mentioned that we are gonna throw this monochromatic light, the light which is of one color. So this was for just one mark. Then we can say that we need to calibrate our calorimeter by using distilled water. If we are using distilled water and our absorbance is zero, that means that yes, our calorimeter is working all fine. Right. Onwards now, let's have a look on the other question. Now he's saying that in order to determine the concentrations of glucose in potato juice using glucose assay, a calibration curve needs to be produced. So now they're asking us to produce a calibration curve. And this involves using glucose assay to obtain absorbance readings for standard glucose. Now, they are providing you with a 2% stock solution of glucose from which to prepare a range of standard glucose solution. State the glucose concentration the student could use to produce calibration curve and describe how 20 ml of each solution should be prepared by proportional dilution of the 2% stock of the glucose. And you may use a table to show your answer and glucose concentrations to use. So definitely, we have 2% glucose in our start. The glucose concentrations to use, say we are going to make, we have 2%, and then we are making 1%, and then we are making 0.1%, and then we are making 0.25%, right? So this is number 1, this is 2, this is 3, this is 4, and let's say we are also making 0.125%, and this is our 5th reading, right? So now, how can we prepare these readings? How can we prepare them? We need to talk about it. So we need to make this table, or we can use that beaker thing that we are used to of doing in our practicals. For example, this is our stock solution, which is 2%. And then we'll take a test tube. Now we are going to make 1% of it. So take 10 ml of this 2% stock solution in it and add 10 ml of water in it. So this is 1%. Now take another tube, take 10% of this 1% in here and add 10 ml of water. Now this will make it 0.5% and so on. This is how you can make these 5 concentrations. Now it's asking you that sketch on a graph of the expected calibration curve that student would obtain and include the axis labels. So on x-axis, we are going to label percentage of glucose concentration.
Sir, I didn't understand the dilution part.
Dilution part समझ नहीं आया? उसने कहा कि आपको 5 dilutions बनाने, आपको जो given है वो हमारे पास 2% है. 2% से 1% बनाने का तरीकाकार क्या है कि आप इसके अंदर आधा 2% लो जो कि हमने 10 ml लिया और आधा पानी डाल लो. जब हमने इसमें आधा पानी और आधा ये लिया तो ये 2% से क्या बन गया 1%. अब अगला वाला 0.51 का भी हाफ है. अब आप 1 का लोगे 0.10 ml ये 1 वाले का और इसमें 10 ml वाटर डालोगे तो अगेन ये हाफ हो जाएगा, ये 0.5. अब 0.5 वाले का 10 ml लोगे और इसके अंदर 10 ml वाटर डालोगे तो ये 0.125 बन जाएगा. Do you understand it now?
सर वाटर की कंसंट्रेशन सेम है, ग्लूकोज की कंसंट्रेशन ये है 2%, 1%, 0.5 एंड अप टू 0.125.
हाँ जी. ये बनाने का तरीकाकार क्या है, एक मरतबा फिर देख लें. हमारे पास एक सोल्यूशन है जो कि 2% है, ये हमारे पास इस टेस्ट ट्यूब के अंदर मौजूद है. अगर हम एक और टेस्ट ट्यूब लेते हैं और उसके अंदर 10 ml ये वाला 2% डालते हैं प्लस इसके अंदर हम 10 ml वाटर डालते हैं, तो मुझे बताओ इसकी पावर बढ़ जाएगी कि घट जाएगी?
कम हो रही है, शायद
कम हो रही है ना, कम हो रही है. आधा पानी डल गया तो ये 2% से 1% में कन्वर्ट हो गया. अब इस 1 का भी अगर हाफ करना है तो क्या तरीकाकार? अगर हमने इस 1% का भी हाफ बनाना है तो क्या करेंगे?
मतलब उसमें और कम कम डालते जाएंगे।
जी, हम अब 1% का 10 ml उठाएंगे और फिर इसमें 10 ml और पानी डाल देंगे। ये 0.5 बन जाएगा। अगर इसको भी मजीद
सर
डाइल्यूट
सारी
करना है,
डाइल्यूशन इसी तरह होती हैं?
ये सारी डाइल्यूशन इस तरह होती है। ये जो हम डाइल्यूशन कर रहे हैं, दिस इज कॉल्ड अ सीरियल डाइल्यूशन, इन विच जो हमारे पास रिजल्टेंट चीज बन रही है, इट इज डाइल्यूटेड बाय फैक्टर ऑफ हाफ। जो हर अगली डाइल्यूशन बन रही है वो 50% डाइल्यूट होती चली जा रही है।
ओके ओके।
अब तीन तरह की डाइल्यूशन होती हैं। उसके लिए आप मुझे याद करवाइएगा, मैं आपको प्रैक्टिकल के सॉल्व पेपर्स भेजूंगा, उसमें मैंने डाइल्यूशन बहुत डिटेल में समझाई है। अच्छा, अब हमारे पास एक्स एक्सिस में परसेंटेज ऑफ ग्लूकोज कॉन्सेंट्रेशन आ जाएगी, और वाई एक्सिस पे एब्जॉर्बेंस। अब क्या होगा हमारे पास, हम क्या एक्सपेक्ट करें? एक्सपेक्टेड कैलिब्रेशन कर्व कैसे आना चाहिए कि जैसे जैसे ग्लूकोज की कॉन्सेंट्रेशन बढ़नी चाहिए, हमारे पास एब्जॉर्बेंस जो है वो भी बढ़नी चाहिए। ग्लूकोज की कॉन्सेंट्रेशन बढ़ेगी तो हमारे पास एब्जॉर्बेंस भी बढ़ती चली जाएगी। अगला क्वेश्चन देखिए, वो कहता है द स्टूडेंट डिसाइडेड टू इन्वेस्टिगेट द इफेक्ट ऑफ स्टोरेज टाइम ऑन ग्लूकोज कॉन्सेंट्रेशन ऑफ पोटैटो। तो ये देखना चाहता था कि जो इन्वेस्टिगेशन का टाइम है वो कैसे ग्लूकोज कॉन्सेंट्रेशन को अफेक्ट कर रहा है। वो कह रहा है डिपेंडेंट वेरिएबल बताएं इसका क्या है। डिपेंडेंट वेरिएबल है ग्लूकोज कॉन्सेंट्रेशन, और इंडिपेंडेंट वेरिएबल है स्टोरेज टाइम। क्योंकि जैसे जैसे हम टाइम को बढ़ा या घटा रहे हैं, ग्लूकोज की कॉन्सेंट्रेशन अफेक्ट हो रही है। सो इट इज डिपेंडेंट अपॉन टाइम, दिस इंडिपेंडेंट वेरिएबल। अब आगे वो कहता है स्टूडेंट वाज प्रोवाइडेड विद फ्रेशली हार्वेस्टेड पोटैटोज, स्टैंडर्ड लैब apparatus एंड अ कैलोरीमीटर। Describe a method using a glucose assay that student could use to investigate the effect of storage time on glucose concentration of potatoes. Your method should be set out in a logical order and be detailed enough to allow another person to follow it. और ये जो ऊपर हमें एसे वगैरह की डिटेल्स हैं ये इंक्लूडेड नहीं होनी चाहिए। अब इसके अंदर आपको क्या क्या चीजों का ध्यान रखना है। वी हैव टू अटेम्प्ट दिस क्वेश्चन फॉर 6 मार्क्स। नंबर 1, आप अपनी इन्वेस्टिगेशन में सेम पोटैटोज यूज करेंगे। इन पोटैटोज की सेम स्टोरेज कंडीशंस होनी चाहिए। यानी सेम टेम्परेचर, सेम लाइट, सेम कार्बन डाइऑक्साइड कंसंट्रेशन एंड सेम ह्यूमिडिटी। अब आप इनको डिफरेंट स्टोरेज टाइम्स के लिए प्रोजेक्ट करेंगे। डिफरेंट स्टोरेज टाइम्स, फॉर एग्जांपल for 1 week, for 2 weeks, 3 weeks, 4 weeks, whatever. You have to tell about the time. Then, for all these values of time, you need to use the same method for juice preparation. इस जूस की प्रेपरेशन का मेथड भी सेम होना चाहिए. और जब आप ऐसे परफॉर्म कर रहे हैं तो वॉल्यूम ऑफ जूस भी सेम होना चाहिए. Volume of juice that you are using for essay should be same. उसके बाद for each storage time you guys need to measure the absorbance. Absorbance. And then what you need to do is you have to perform all this experiment 3 times and take an average to make it reliable. Then you also need to talk about the risks. So here, let's talk about a couple of risks. The first risk is Benedict reagent. And this Benedict reagent may be irritant. And what is precaution? Use gloves or eye protection. Then, when you're using hot water bath, you can include any two of these precautions. When you're using hot water bath, don't put your fingers in it. It may burn your finger. So use holders. When you're cutting this potato and you're using a knife or a surgical blade, it can result in some injuries. So cut it away from your hand. So that was half part of this P5. Now for the next half part, we need to wait for another day. And then we will discuss what statistics are involved in the other half part, which is not very difficult. So let's call it a day. I'm going to stop this recording.